So does everybody have your copy up here? Okay, and you should have a copy for yourself too. Okay, what we're going to do this morning is we're going to do an introduction to the lab you're going to be doing this week. There's several things you've got to keep on task, and there are some things that you should be aware of, even if you have read the lab handout, which hopefully you have. Then what we're going to do is we're going to give you the reading quiz, and then after the reading quiz, I'm going to ask you to get together with your partner and go through your lab procedures and compare them and come up with a common lab procedure that you're going to use. Okay? And then you can leave after you're done with that. When I look at these this morning, basically what I'm going to do is I'm just going to grade these based upon, did you make an honest attempt to do the assignment? Okay, you might not be right. Okay? One of the things that I would like you to do when you come up with a combined lab procedure is that you should only be working with one piece of dialysis tubing in that model cell membrane experiment. Okay? Some of you are doing multiple dialysis tubings you don't need to. And so that's something you can talk about with your partner, saying, how can we accomplish this particular lab's goals with a single dialysis tube and those two solutions, A and B, okay? And then it's important to remember that you've got to answer the following questions in your experiment. Is the membrane permeable to water? Is it permeable to glucose? Is it permeable to protein? Is it permeable to iodine? Is it permeable to starch? Okay? So there's 5 things that your experiment's got to address in terms of being able to collect data to determine its permeability. And you will have to use techniques you learned in the first lab in terms of tests to be able to detect those molecules of glucose, protein, and starch. Okay? So that's where we'll take into. All right. So we're going to be doing 3 different experiments today. One of them has to do with determining what environmental factors influence beat membranes and how those particular parameters of temperature might put those beat membranes under stress. So that's one set of experiments we're going to do. Another one is the model cell, which you're writing up the procedure for right now, or have done. And then the third one is a really cool experiment on transport in yeast. What's the first one you need to start with? The first one you need to start with is the beat experiment, because that particular experiment takes some time for the bead tissue to become exposed to the different temperatures so you can determine whether there is stress on those particular membranes. And it gets a little bit confusing because there are kind of 2 exposure regimens that you've got to go through with different time periods depending upon the temperature of exposure. So, one of the first things you need to do is you need to make your beat cores. And we'll just have little beats there. We'll have a cork bore. Has everybody seen a cork bore before? It's just a little tube with the handle on it that's hollow inside, and you push it through the beat, and then inside there's a little piece of beat tissue, and you can use a stick to then push that out. You're going to cut those to a particular length. The diameter of all those will be the same because the cork bores all have the same diameter. And one of the things that we ask you to do initially is, once you've got those particular segments cut into their particular lengths, that you put them in the top of a little plastic petri dish, put a little bit of tap water in there, and just rinse those particular bead segments off. You may know why we're doing that? Why do we need to rinse the bead segments off? Yeah.
You remove the color release after cutting it.
Yeah, when you cut the tissue, that's going to tear open cells. And what we're looking for is at these different temperatures, are membranes damaged? And if they're damaged, is that purple beta-cyanide pigment released from the tissue? Well, if you're cutting the tissue, that's going to damage the membrane. So we want to wash off that pigment that's leaked out because of damage. So then the pigment that leaks out later will be because of the temperature effect on the membranes, not the cutting. Okay, so we've got several temperatures, and the 2 that go in the coldest temperatures, -5 and +5 Celsius, you're going to put the bead segment in a test tube with no solution in it, and you're going to put that in that particular temperature of freezer or refrigerator for 30 minutes. You're just going to let them sit in there. Okay? You're going to take the other 4 bead segments and put them in water of different temperatures. So we'll have some water baths sitting out, and they'll have different temperatures. There'll be a thermometer, and they're probably labeled, aren't they, as to which they are. So what you're going to do for that exposure is you're going to dip out a little beaker of water, and you're going to throw the bead segment in the water. Okay? But are we going to leave it in there for 30 minutes? No, we're going to leave it in there for a much shorter period of time, 2 minutes. Okay? Why a shorter period of time when they're soaking in the water as opposed to the air? So that seemed to kind of violate, the idea that everything is to be the same in the experiment. The efficiency of heat transfer between the environment and the tissue is going to be much lower, much slower, when they're just sitting in air. Okay, if you go out and sit in 100-degree temperature on the mall, it's going to take a while for your body to warm up just because of sitting in that air temperature. But if you go into water of the same temperature, the heat transfer is going to be much greater and much faster. Okay? So we need a shorter exposure for the 4 water temperatures and a longer exposure for the air temperatures. Okay? After that time, either 2 minutes or 30 minutes, respectively, you're going to pull those particular segments out. And you're going to put them into a tube of 10 mils or 5 mils of tap water. And you're going to let them soak for 30 minutes. So what are you doing now? If there has been damage to the tissue by exposure to that temperature, then the pigment's going to start leaking out. In 30 minutes, some of that pigment will have leaked out of the tissue into the solution. And the solution will start to change color. It'll get redder. So the more damage, the redder the solution will be. Okay? Once they have been in this solution of water for 30 minutes, then you need to pour the water out into a clean test tube or cuvette. And then take an absorbance reading. It doesn't really matter after you've poured this solution off of the beads when you take that reading. Because the beads are no longer in it, they're no longer leaking. Okay, so the color of this will then not change any longer. So you can take these spectrophotometer readings whenever it's convenient around one of the other experiments that we're doing. Now what you can't forget to do is pull those out of the refrigerator and freezer at 30 minutes, put them in their tubes of water for 30 minutes, and then take the absorbance readings of those as well. Does that make sense? The water treatments, 2 minutes in water, pull the bead segments out of the warm water, put them into room temperature water, 5 mils, let them sit for 30 minutes, pour off the liquid, and then read the absorbance at some point in time. For the 2 air temperature ones, 30 minutes at that temperature, then water, soak for another 30 minutes, and then pour off that water and read its absorbance at some point in time. And I'm sure you'll probably give them a heads up, right, to watch out for the 30-minute exposures.
Yeah. You can see right away why it was really important for you to read over that lab protocol carefully, because this is just one of 3 experiments we're doing. Conceptually, what you're doing is taking bead cells, all right, and they have membranes, phospholipid bilayers. You're exposing them to a range of temperatures from minus 5 to 75 degrees Celsius. So that this is our independent variable, okay, and we're looking at the effect of different temperatures on the integrity of the membrane of those plant cells. Okay. What 5 degrees, or what's minus 5 going to do to them compared to 75 degrees Celsius, right? What we're then going to measure, our dependent variable, is how much pigment leaks out as a function of the different temperatures. So this is our dependent variable, or our independent variable. This is our dependent variable. How are we going to quantify the amount of leaked pigment? We're going to use spectrophotometers. Remember last semester, things that have color we can measure using a spectrophotometer. The pigment, right, is a deep red color. So we can measure it because it has it has a wavelength of light that it absorbs efficiently. Okay? So this is just experiment 1. And there's, it takes basically an hour to run the whole experiment. Okay? But while this is running, then we go to...
Well, let me say something else about experiment 1. Once you've got your data, the groups, each of the groups are going to put their absorbance readings up on the board. And so we'll have all these temperatures, -5, +5, etc. And then we don't have this many groups this semester, but each group will put their absorbance reading in the appropriate column for the appropriate temperature. Okay? So what are we going to do with that? We want to now see is, are there any real or significant differences between the temperatures of exposure and what's happening to those beads? So what are we going to need to do to answer that particular question? Well, you're going to have to be able to calculate a mean for the class data for each of the temperatures. We're going to want to know a standard deviation, because standard deviation is going to be necessary to answer that question about whether any of these mean differences are actually significant differences. Remember that from last semester. You can only draw conclusions about data based upon differences that are statistically significant. Okay? So, the next step you'll have to do with this is you'll have to write down the class data and then enter it into your old friend stat book from last semester. Okay? So when you do stat book, it's very important to remember that you retitle your column headings in stat book from control, experiment 1, experiment 2, etc., to the temperatures. Okay? So you're going to make a stat book, open up stat book and start entering the data, and then you'll just transpose the class data to each of those temperatures. And it will calculate your mean and your standard deviation and give you your n value. Okay? So, if you look at this data just at the face, it looks like, wow, there's a real difference between exposure at freezing and refrigerator temperature. But in order to draw that conclusion, we need to ask the question, is this difference statistically significant? Okay? Is this difference statistically significant? So we need to compare each of these means and see, is there a true difference? What is the least level of confidence we are willing to accept to say things are statistically different? We want to be 95% certain at least. If we're more certain than that, that's even better. But the minimum level of statistical significance we'll accept in order to draw conclusions is 95% confidence. So you put that data into stat book, and that'll give you that type of information. And then remember, up at the corner, you can click that little T-Calc button, and that will take you to this data chart. And remember how this is set up. This allows you to compare the mean values of any two temperatures. So if we want to compare the mean of the -5 temperature and the +5 temperature, this is the t-value for that particular comparison. Do you remember how you determine whether this t-value indicates these means have a significant difference? What do you compare that t-value to? You need a t-table, right? And if you look at that t-table, there are rows and columns. The column you're going to want to look at is the 95% confidence level. And the row you're going to want to look at is degrees of freedom. Do you remember how you calculate degrees of freedom for comparing two sets of data? Do you remember the formula? Degrees of freedom is equal to n-1 plus n-1. What's n? N is your sample size. In our particular case, the number of groups. So if I want to compare -5 and +5, then my n value would be what? right? 12. So I would look at the row that says 12 degrees of freedom. I would look at the column that says 95% significance. And that would give me in that intersection an expected t-value. And if I compare this calculated t-value, does this have to be bigger than or less than the expected for the difference to be significant? Remember? 50-50 chance. This calculated t-value has to exceed the tabular t-value. Okay? So what you'll sometimes find is that there will be means from different data sets that look like they are very large, and you just look at that and you say, well, that's different. But if you do the t-value comparison, it might not be. That's particularly true if there's a lot of variation in the data. If the group's numbers that led to this particular mean were all over the place. And so that's one of the things that's interesting to look at when you start collecting your data is how much variation is there in the data. Look at this column over here. One group got 1.4. One group got 0.25. That's a big difference, right? So the bigger the spread is in these numbers, the bigger that mean difference is going to have to be for the difference to be significant. If these were all just right dead on with one another, then a very small mean difference might be significant. But if the data is all over the place like this one is, even numerically means that are pretty different from one another aren't significant because of all the variation in the data. This is something we're going to be doing several times in lab. And so if you're having trouble with knowing how to put data in the stat book and interpret the results, you've got to get some help with it. Okay? Austin can help you. Either one of us can help you.
Do you have a slide on the t-table?
I thought I did, but I don't. But they've got one in their lab packet.
Okay, when you are looking to find out what the t-value is for determining significance, the table doesn't put 95% confidence at the top of the column. It has a p-value. And we never really explained necessarily how that p-value is calculated, but to give you an idea, if you want to be 95% sure that they're different, that means there's only a 5% chance that they're not different. So another way of saying that you are 95% sure they're different is to say there's only a 5% chance that they're not different. That's what the p-value is. So you want the p-value, if we're going with 95% confidence that they're different, you want the p-value to be no greater than 5%. Now they don't put percent on the top of these columns. They put the decimal equivalent, which is 0.05. It's the same as 5%. So you want the column, if you want to say that it's 95% different, you want the column where p is equal to 0.05. Okay?
Take the left-hand column. The other one is 0.01, which is 99% significant.
Yeah, I don't know. I have to look at the column.
Yeah, I think there are two columns.
Okay.
So that's something you will have to do afterwards, okay? You're probably not going to be able to get this done by the end of the period because everybody's got to get their data up on the board. But you can come anytime into the lab and put your data into StatBook and then get your means and get your t-value comparisons. And by the way, have they downloaded something? Okay. If you want to work in the lab too, you can print for the lab in free. Okay, so I know some of you said you're running out of printing money. You can print lab work in the lab for free. Okay. Okay, we've already talked a little bit about the permeability properties of a model cell, and so once you get the bead experiment set up and get everything sitting in their appropriate tubes, then you can start the dialysis tubing experiment, okay? And you're going to double check that with Dr. Morden at the beginning of lab after you've run through it with your partner to make sure you have set up your experiment correctly and collect the data that you need to collect. So I think that's The third experiment you can then start once the dialysis tubing is going is movement of substances into and out of yeast cells. And in this particular experiment, we're actually going to be working not with a model cell, but an actual cell, a yeast cell, a fungus. Okay? And one of the things we're going to be able to do by doing some simple manipulations is to determine some permeability properties of yeast cell membranes. And we're also going to be able to answer an interesting question: is the cytoplasm of a living yeast cell naturally acidic or basic? Okay? And that's actually going to have some relevance in an upcoming lab, whether that's acidic or basic. Okay, here's some of the substances you're going to be using in this experiment. One is a dye. The dye is called neutral red. And this red dye is a pH indicator. Y'all know what pH indicator indicates? Ever worked with litmus before? Sometimes you use litmus in chemistry, and litmus changes colors depending upon the pH. So does neutral red. At an acidic pH, neutral red is a reddish color. At a basic pH, it has an orange-yellow color. And one of the things we'll do initially is we'll just have you put some neutral red in acidic solutions and basic solutions, so you've got a way to compare those colors to make sure you know what they are. Okay. Whenever you are writing down colors, and that's your main data you're collecting in lab this week, are color changes. Ask yourself this question: is the solution or the cells a reddish color, or are they an orange-yellow? So if it looks pink to you, it's considered reddish. Okay? So it's really one of two choices. Is it some form of red, or is it some form of orange-yellow? Okay. Another solution we're going to be working with is sodium carbonate. Sodium carbonate is a base. So if you add neutral red to sodium carbonate, it's going to have that orange-yellow color. We're also going to be working with acetic acid. Acetic acid obviously is an acid. So if you mix neutral red with acetic acid, it'll turn red. In this experiment, we're going to be asked to filter some things. And the reason we're going to filter is because we want to determine colors of solutions and colors of cells. The solution will go through the filter paper into a test tube. The cells will be the stuff trapped on the filter paper. Now, we're going to start out with some fairly large volumes. I think we have like 25 milliliter volumes. And it's not necessary to filter every drop of that. What we can do is just pour some of the solution through the funnel with filter paper inside. And then as long as you can collect some volume that you can see the color of, and you can have some cells trapped on the paper, that's sufficient. Okay? Do you all know how to make a filter paper cone to fit in the filter? Okay. Dr. Morgan will show you. Okay. The other thing that we're going to do is we're going to ask you occasionally to acidify solutions. And that involves adding acetic acid to them. Acidification in our case is going to involve just adding an equal proportion of acetic acid to whatever we want to acidify. So it's okay to just eyeball it. Okay? So if I've got a little bit of solution in the bottom of a test tube, I'm just going to ask you to add a little bit of acetic acid, so you just basically double the volume. Okay? Now, I don't know, do we even have this written out in the lab as a narrative anymore? No. Okay, we used to have the instructions written out as a narrative, and instructions for experiments sometimes are easier to follow in the form of a flow diagram. So your procedure is going to be in the form of a flow diagram. So what we're going to do is we're going to be working with two flasks, Flask A and Flask B. And these give you the steps, and then this tells you what you do next, the arrows. Okay? Notice that there are numbers associated with some of the steps. Those numbers correlate to this data chart right here. So the number 1 right here correlates to this step right here for Flask A, this step right here for Flask B. So basically you're going to do the procedure, and then it's going to ask you to collect some data. For example, what's the initial color of the suspension in Flask A when you mix a gram of yeast and 25 mils of sodium carbonate? Okay? Look at the color. So your data right here is basically going to be colors recorded for Flask A and B. The next step is we're going to boil it in this case. Here we're not going to boil it. So what's going to be the difference in terms of the cells in flask A and flask B after this first step? Well, we haven't even added cells to this step, have we? Are these cells going to be living or dead after we boil them? Yeah, that's going to kill the cells. One of the things you'll see over here in this procedure is there's no boiling. So flask A basically shows you what happens to dead yeast cells, and flask B shows you what happens to living yeast cells. Okay? So you just follow through step by step. Here you filter some, you save some. The stuff you saved, you acidify. So just follow this step by step and then record your information down here. Now, some of these ask questions, okay? Did neutral red enter the cell? How do you know that? The data you've collected will allow you to deduce that particular answer to that question. So depending upon the color changes that have occurred, you should be able to logically figure out whether neutral red is inside the cells. You'll also should be able to figure out, if it did, what was the process. The processes that we're going to be considering are diffusion and active transport. Those are the two kind of outcomes potentially that we could get. Okay? We can determine whether acetic acid entered the cells. The same thing for flask B. Now there's some questions here that don't apply because we didn't do that particular step in that particular procedure. So if it's X'd out here, you don't have to answer that. And then finally, you can, based upon all this information, determine the answer to this big question: is the cytoplasm of a living cell acidic or basic? Explain your answer based upon your results. So this is pretty cool because you can now determine by your experimental results fundamental questions about the properties of yeast cell membranes, and also a fundamental answer to a question about what's the nature of the cytoplasm of a yeast cell, acidic or basic. All by mixing a few chemicals together with yeast cells, doing some filtration, and then making some observations. And so this is a great exercise in deductive reasoning. Make sure that you understand what's going on with this experiment. And don't just let your lab partner reason through it and you write down the answers. Because I can tell you definitively on your first lab quiz, there will be a question similar to this where we'll give you a similar type of procedure, but we'll give you some different results than you got. And then you will have to answer these same kind of questions based upon the results that you're given. So you need to know how to logic through this and understand why the answers to these particular questions are correct answers. And if you have problems with that, we can certainly help you out. I don't think I was going to show the spectrophotometer. We can talk about the spectrophotometer. There are two kinds of specs, and so that's the difficulty, is that there are two kinds of spectrophotometers you'll be using. And each one has a little bit difference in terms of controls and where they are and things. Okay? You want to go?
No.
Okay. So let's take your quiz, and then when you finish your quiz, as soon as your lab partner has finished their quiz, get together and come up with a synthesized procedure among the two of you. You don't have to turn that in. Bring that to lab. Okay?
Don't forget that there will be a short quiz at the start of lab on what we're doing today, and you can use paper notes on the